So our next speaker is Lucienne Ronco. I first met Lucienne Ronco in Marseille, I think, when she was working for Fulcrum. I remember her not only because she has a French first name, but also because she seemed very attentive to patients' voice. And she has now the important role of chief scientific officer for FSHD Society. And she can share with the FSHD community the skills she acquired during her impressive career. She had been in research, management, pharmaceutical companies, and so on, and she will be of great use for our community. And she will speak about clinical trials in FSHD and therapeutic development, completing was what we heard yesterday from Alexandra Belayev.
Thank you very much for a very kind introduction. I'm so grateful to be able to work in this space with the FSHD Society. I'm grateful that June and that Mark Stone came and asked me to step into this role. It's such a pleasure to take everything you've learned and be able to work on behalf of the patients with everything you've learned, and it's been a joy, really. And I want to tell you that the International Standard of Care guidelines weren't just presented to you this morning and prepared for publication, but they were presented at our science meeting that happened for 2 days prior to this meeting, because we have to educate the scientists and we have to educate the clinicians as well. And I was asked by the organizers to give you a very few highlights from the science meeting. One of the highlights was the presentation of the guidelines. And I have to say, I chose the exact same two things that Nicole and Raj chose, which was be fit and also mental health. These are two exceptionally important things that patients and their families can do for themselves and for each other. And there's guidance that's emerging about how to consider the being fit question. A scientist presented on a concept of start low, go slow. And this means you don't go out there and lift 50-pound weights the very first time you're lifting weights, or walk 20 miles or 40 kilometers the first time that you walk. Start low, go slow. If you find yourself out of breath, you've worked too hard, because you are pushing yourself aerobically and your muscles are stressed by that. So I'm not the expert here. I just listened to a talk 2 days ago, but there is expertise that will be available. The Society, and I imagine FSHD Europe, we're going to interview the scientists and put those interviews online so that the information is available. And I have to tell you, the most exciting, not the most important, the most exciting presentation was given by Avidity, which is a company in the United States who's taken the first gene modifying therapy into patients. And the reason it's exciting is they have seen signs that the drug is hitting its target, and they see changes in clinical function for patients. It's very exciting. They followed, I was at Fulcrum, as you heard, for the 5 years that we worked on FSHD, and they followed the Fulcrum guidelines pretty closely, which was nice to see, but also gave us more information about how those endpoints work in the clinic, and they saw benefit in multiple ways across everything they've measured. So I think that gives us confidence that we can reduce DUX4, we can reduce it enough that can affect patients' outcomes, and I'll be showing slides in more detail about each of the therapeutic approaches. The other interesting thing we saw from Beatrice Bifarale is that we know DUX4 is present in muscle. What does DUX4 do? It turns on genes, and I'll talk a little bit about that. It turns on an antiviral pathway. So when you get a virus, your body responds by turning on proteins that then go to your infected targets, infected organs, and it drives a very strong interferon response. Well, they have found, Bifarale and group has found, they're in Italy, that muscles start to make interferon when DUX4 is there. What happens in an animal model? They ask the question, if we use an antibody or some way to block interferon, it turns down fat infiltration, which happens in our muscles because of DUX4, and fibrotic behavior. That's very interesting. That suggests that an antiviral normal response in our body may be exacerbating or making the muscle disease worse. And we can stop it. There are approved medicines for interferon already available. They are not immediately going to be available for FSHD, but it gives us many good questions to ask. I want to talk about, this was very exciting. There's the first description of FSHD by the group here in Leiden in Africa. And we're challenged to describe FSHD in Africa because there's not a great quality of health care across the continent, but we're starting to understand it's there. It looks a lot like FSHD in other continents, and it's valuable for us to understand that, and it's valuable for individuals living in Africa with FSHD to have access to diagnostics as well as to treatment. This is something I'm very excited about because it was developed by a scientist clinician that I worked with at Fulcrum Whole Body MRI. If any of you have been involved in the clinical trials, you may have been asked to slide into an MRI machine, have your whole body muscles imaged. A woman from the University of Virginia, Sylvia Bleckner, is now taking artificial intelligence, machine learning, to the images that Fulcrum and others have collected, and she's finding exceptional things out about FSHD, things that we could not tell previously without the imaging. Because now we have an opportunity to look at almost every muscle in the body, and the value of that is huge. So a lot is going to come out over the next few years. Let's see if I have any other. I just want to tell you about the poster that won our poster award. It was called Estrogen Rescues Muscle Regeneration Impaired by DUX4 in a humanized model in mice. And that's exciting and interesting because we've always known that there are subtle differences between men and women in how they progress, and that estrogen may have a protective effect on the muscle. And it's protective against diabetes, it's protective against Alzheimer's. So we understand that estrogen can be very protective and maybe in muscle disease as well. So please come and ask me any questions about what I just talked about the IRC after we're done today. But now I'll start talking about the slides that I've put together. So I'm with the FSHD Society, but our intention is to really work globally and as a community to drive all of this new knowledge that we have forward for our benefit. This first slide has a lot going on, but this is called a Drug Development 101, a preliminary course. And you really have to understand, because FSHD is a genetic disease, which you've heard about. You have to understand just a little bit about the disease, and I know you've seen this before. The defect that causes DUX4 expression is in our DNA, and it impacts the production of this protein, but there are steps to get to the protein. We make RNA, we make early protein, then we make well-folded protein, and then that goes on, as I was telling you, to activate an immune component, and then it drives damage in the muscle cell and death, and then damage and death in the muscle. We start to get fat coming into the muscle. We start to get fibrosis in the muscle. How many ways then can we attack this disease? Can we turn off DUX4, and can we help the muscle to regenerate? We have a lot of ways, which is awesome. We can work to repress DNA by remethylating the DNA that is upstream of DUX4. And if we remethylate it, if we epigenetically repress it, we can turn off DUX4. It's never going to even be expressed. That's a very powerful way. We can knock down DUX4 mRNA once it gets made with oligos, with siRNA, and again, I'll talk through each of these examples. We can block or remove the DUX4 protein. There's a technology called a degradome, or degrading, and we can target the protein to be degraded. That's a process, that's an active body of work. We can suppress inflammation or your antiviral response, and I talked about that a bit, but there are multiple ways to do that. I'll speak more about that. We can try to drive muscle building. Myostatin blocks muscle regeneration and muscle building. So if we block myostatin, we can regenerate. I'll talk more about that. And then we can stimulate muscle growth. I won't talk much more about that. We're in early stages, but there's multiple ways to think about doing that. And then something that should give you great hope, gives me great hope, is that we have many approaches. We have preclinical approaches that are actively ongoing with all of these companies positioned preclinically, meaning they're doing the work in human cell lines and in animal models to understand if they have an approach that's going to work to reduce DUX4, to reduce inflammation, to enhance muscle growth. We've got EpiCRISPR, they're doing the epigenetic repression, but others are as well. Arrowhead Pharmaceuticals, together with Sarepta, and then companies like Avidity, Roche, and others who are rapidly moving to phase 2, and honestly, Avidity should be popped down to here because they announced just last week that their phase 3 has begun. That's very exciting, and I'll talk a little bit more about that. I won't spend too much time here except that it's a long path, and I know you've all heard this and you're tired of hearing it, but it's a long path to the clinic. All that preclinical work, the clinical research, clinical trials, and then between our finding of success here and launch, sadly, there's a lot of work that needs to be done. You need to make the medicine, and if the medicine is complex to be made, like the Avidity molecule, it takes time. And before you put it in a human, you want it to be high quality, and that takes time. So there's a big step between finding efficacy here in a clinical trial and launch. Now, I won't talk about that today, but I intend to get Avidity and Dyne and others to come together and answer those questions so patients understand. I won't talk about this at all, honestly, but we have many phases of clinical evaluation. And I'd rather get right to the medicines. So I've skipped the Fulcrum trial, but I included it at the end because I think there's something to be learned there. But I went right to Avidity and the Fortitude study that they reported just last week and presented the science for it at the IRC meeting on Friday. This is an approach that removes the RNA for DUX4. And when I say it's a complicated therapeutic, it is a complicated therapeutic. It has multiple parts. It's an antibody oligonucleotide conjugate that targets DUX4 for degradation. The antibody is essential for targeting the molecule to the muscle. Every one of your muscles has something called transferrin on its surface. This antibody sees transferrin, binds to it, gets taken into the muscle cell. Then there's a non-cleavable linker, but there's an siRNA molecule that then is released, and it binds to the RNA from DUX4, and it degrades it. Boom, DUX4 is gone. It's a very nice approach. It must be administered by IV administration, and depending on how tall you are and how large you are, the administration can take 2 hours, 3 hours, something like this, but it's effective. And the Fortitude study, which is now complete, had very specific participants, and they mirrored the initial studies with reachable workspace, with being able to walk 10 meters, and with MRI, all exceptionally valuable for their ability to understand if the drug is working and for our ability to further understand the disease. So it's completed, and the data was reported, and I'm just going to go right to the data. Dr. Jeffrey Statland, who runs the Clinical Trial Research Network in the US, in Canada, and in Europe, in Brazil, presented this data and showed showed some nice things. And I'll just say, we saw an improvement in the upper limb mobility, which is here, in strength, which is here, and in mobility in a 10-meter walk-run. The 10-meter walk-run is kind of a cool endpoint where you have to walk as fast as you can for 10 meters, and they time you every time you do it. And what you see here is placebo in gray for each of the figures, and Delbrex, or that antibody conjugated oligo, in orange. And improving is moving to the right for these two figures and to the left for this figure, because the shorter time it takes you to run 10 meters, the better. All right, so here, your reachable workspace, which is the endpoint that measures 4 quadrants of upper body activity, because patients have told us that upper body function is very important, and that's why it was initially included in our evaluations. You gain area of ability to move your upper body after treatment with Delbrex, and this difference is what's shown between the gray and the orange. The 10-meter walk-run, again, it's actually going in the opposite direction because you're running that distance in less time. So here's the placebo-treated and with Delbrex treated, and it's going in this direction, taking less time. The change in muscle strength is probably self-explanatory when you think about strength. Again, they saw with a placebo here a shift here to strength. Well, these numbers look really small. How effective, how important are those changes going to be to patients? These are very important questions that we have some answers to for some of these, and I'll speak to it in the next slide. But these subtle changes in a patient population as diverse as the FSHD patient population can be very important and very significant. The quality of life was evaluated as well, where patients in the study were asked, how is your overall pain and health, and what is your impression of the change? Did you have a strong impression of change or not? And in this case, for the pain, improvement moving to the right. For overall health, there was an increase, and for worsening pain, there was a diminishment. So while there is overlap, there is a shift. The global impression of change is also moving in the improvement direction, where they've asked about change and they've asked about severity, and there was an improvement there. And I'm going back to this question, and I took the 10-meter walk-run as an example. What do changes of 2 to 3 seconds mean for a patient's abilities? Well, Dr. Statland has been doing this very careful work to ask that question and to be able to report on it. What he's been able to tell us from the natural history study is that a 5 to 10 second change suggests a person is affected, but that they can move without a device. But if there were an 11-second change, or you ran the walk-run in 11 seconds, suggests that you've required an AFO. 12 to 15 seconds suggests the requirement for a cane or a walker. 21 seconds suggests the requirement for use of a wheelchair. This is hugely valuable to us because now we have to go to the FDA or the EMA and say to them, we see a change of 3, 4, 5 seconds. We know that those changes of seconds reflect a profound difference in the quality of life and how an individual functions. We want you to approve because we can better it by these number of seconds. They seem small, but they're very significant. Okay, now I'm going to talk to some other portion of the avidity presentation, because even after I presented this to the volunteers who raised money for the FSHD Society in the US, they came to me and they said, and they all are patients, what is a biomarker? So I tried to answer that question. It represents the disease process in a patient. A FSHD biomarker, because we want it to represent DUX4, would indicate if DUX4 is present and active. And this is very important. It can come from the muscle, it can come from the blood, or it can come from any relevant tissue. And if it's really valuable, it changes with progression of the disease, so its numbers change as the disease gets worse, or with an efficacious treatment. So here I have a drawing of a muscle, and I put these little things in here that are little shapes and sizes. Each is to represent a protein. DUX4, when it's active, turns on new proteins that we don't usually find in muscle. And those, in part, are what are toxic to your muscle function. We can measure these proteins in the muscle, but those proteins can also leak into your circulation and be detected in blood, and they can tell us about your status or your position of the disease. There is one protein that, together with a long-term researcher in FSHD who's very knowledgeable in the field and very knowledgeable about gene regulation, they identified this protein, KHDC1L. To simplify it, Avidity is calling it CDUX4 because they can see DUX4 in circulation by measuring this protein. It's the name of their new DUX4 regulated marker, and I just called it this one right here. What they found is they can measure it in blood. DUX4 is active. We make these proteins. DUX4 makes muscle cells die and releases proteins, but also this CDUX4 may naturally be released from a muscle cell without it breaking apart, goes into the blood, and we can measure it. Of course, we also can do muscle biopsies, but we hear repeatedly and consistently from patients, please try to stop having to do muscle biopsies in a clinical trial. So CDUX4 is the name of this regulated marker, and with Delbrax, they see CDUX4 reduced after treatment. So this gray line consistently is the placebo throughout the talk, and then the orange line is treatment with Delbrax. These little blue lines show when they treated with Delbrax and how often, and what happened to the circulating biomarker of DUX4, CDUX4, it went down. That's really impressive. First time it's been shown with a therapy that they can see this kind of effect. There's also another marker that describes protein muscle function. It also went down. So they have two independent protein markers in the blood showing that the muscle is making less DUX4 and the muscle may be getting better. So they've ended that study and they've reported on it, and their confirmatory phase 3 design is in place. That's going to be the 10-meter walk run, something else called the timed up and go, which I think a lot of you have experienced. And then as we discussed, the muscle strength test, the quantitative muscle test. They're seeking 200 individuals, ages 16 to 70. They'll be giving the drug every 6 weeks, and they'll be in 45 global sites. It's a huge trial, big undertaking, very exciting. They'll also do the patient-reported outcomes that I've described and CDUX4 measurements. They also received confirmation from the FDA, that's where they went first, that they can take this path called accelerated approval. If CDUX4 continues to show a response to Delbrax, they can move rapidly towards approval. It requires post-approval work as well, but it may make the new medicine available to patients more rapidly. And that's what the FDA, the EMA, would be seeking to accomplish. We have FSHD moving forward, no valuable treatments now. The regulators, if they see the data is strong, positive, and consistent, will consider strongly accelerated approval, and let's watch what happens over the next year to two. So Roche has an approach called Maneuver, that's their clinical trial name, where they are, I told you about myostatin, an antibody that blocks muscle growth. It's a negative regulator of muscle growth. They have an antibody that sees myostatin, inactivates it, takes it out of its active zone, and it should allow muscles to grow. In preclinical models, it does allow muscles to grow, and it's a sub-Q injection. This study is complete, and it should be reported, I believe, in early next year. Well, the date is Q3 2026, but perhaps sooner than that. It was in many places in the United States. Reinforce is another Roche study where they're using an antibody, and I'll just go right to a description of that antibody. It is also fully enrolled and happened in Ottawa, Canada, Nice, France. Reinforce took on one of those factors that drives the immune system to see the muscle and target it, thinking that there's something going wrong there. IL-6 can be seen by a specific antibody to IL-6, and IL-6 levels are elevated and measurable in FSHD patients' blood compared to healthies. It's higher in FSHD people than it is patients than it is in healthy blood, and it plays a significant role in muscle degeneration. It can promote wasting, atrophy, but it can also contribute to muscle repair. So there's a fine balance, perhaps. So this work, I believe, is close to completion, and we should have a report of it within a year. Arrowhead with ArrowDUX4. It's another approach to reducing DUX4. In this case, they have a technology where they can target their DUX4 reducing factor to muscle. They have strong chemistry that facilitates the linking between their targeting molecule and the targeting molecule, sorry. And its goal is to remove the RNA that I described to you that from DUX4. It's also administered by sub-Q. This study design will have around 60 participants, these ages, only of FSHD1, with a specific severity score. They also will do MRI with the intention to do muscle biopsies. Sarepta, which is a pharmaceutical company that has a lot of experience in muscle disease and DMD, DM1, and now they're stepping into FSHD by collaborating closely with Arrowhead on this project, is taking this into the clinic in New Zealand, Australia, Thailand, and you can find information here. EpiCRISPR, I described to you, we're one of the companies that are looking at the very early phases of turning on DUX4. We know that the region with DUX4, when the disease is on, is hypomethylated or undermethylated, and that allows DUX4 to be expressed. Their goal is to remethylate this region with complex chemistry that I won't describe, but it reduces the expression. It's expected perhaps to be long-lasting. Perhaps you only need the medicine once. Its delivery vehicle is a virus, and I'll speak a little more about that. And it's this CRISPR-based technology that's kind of miraculous and very exciting. They will take FSHD1 also. They are already enrolling. They're going to do the 10-meter walk-run and also the MRI. Only 6 to 9 participants because it's a very early use of this technology in FSHD, and they want to ensure they understand the safety before they go much further in taking it into more patients. I just wanted to New Zealand and the US. So these are the places, New Zealand and the US, where this study is being done. It'll be a small number of individuals. And now I just wanted to go back to the Fulcrum study. Why is it important to us? It's done. We don't think it worked very well. Why do we care about it? Well, it's taught us a lot. What can we reliably measure? Is reachable workspace going to be useful going forward? Is it not? Does the Ricci score describe patients well enough that those are the people who should be included in the trial or not? And how does the MRI help us to understand the disease? And is it valuable for us to do MRI in clinical studies? So Fulcrum started these. I believe, this is my strong bias, the MRI imaging is exceptionally valuable because you could have a clinical severity score of 3 and you could have 4 muscles, one here, one here, one here, one here, affected. You could have a severity score of 3 and you could have 12 muscles that are affected all over your body. So the severity score is not valuable in telling us the current positioning or current status of a patient. So these are the kinds of things we learn from running a clinical trial. So never think that you were involved in a clinical trial, it wasn't successful, it failed, and I shouldn't have done it. Think instead, wow, I contributed to all of that knowledge. And that knowledge is going to make the next clinical trial successful. That's hugely valuable to us. So many people are contributing to this work. Again, you should find hope from this slide. More than 12, more every day we're being contacted, the society is being contacted by. And I couldn't walk away without telling you at a very high level the potential benefits and risks associated with every new therapy. Because for sure, there are potential benefits and risks. There are many approaches as I described. So Fulcrum had a small molecule, antibodies, conjugated oligos, and viral carried or viral delivered gene therapy. Let's start with small molecules all the way on the left. They're easy to give because it's a pill. You take it, and you get to take it at home. And because it's a pill and it goes into your bloodstream, it could reach every organ, every muscle in your body. There's a lot of potential benefits there. But the risks are that you could have toxicity from this new drug, and if it reaches every organ, it could have an unintended consequence on an organ it's not supposed to have reached. So there's risks, benefits, and risks for every approach. Antibodies, highly, highly specific. That's beautiful and very nice, and we've already used them in many cases to treat many diseases. So there's a good long history of the use of antibodies in therapeutics. That makes us comfortable too, because we've learned a lot about their safety. Risks. You can have immune reactions at the ejection sites, and you can have target-specific effects, like when you reduce IL-6, is it all positive, or is there some negative component to it? Does the positive outweigh the negative? Those are questions we have to ask clinically. And of course, there's the frequency. You might have to go every 6 weeks for an administration. But if you have FSHD, you may be willing to do that, but you've got to be close enough to an IV site to get it done. The conjugated oligos delivery is by a similar method to the antibodies. It can be very specific targeting that makes it very nice. And we've seen benefits in other muscle dystrophies, and that gives us hope that this approach is valuable. But the risks are you've got to have frequent administration, and if you go to too high a dose, there have been high dose dependent issues. All the companies that are working in this space are working to find the most efficacious and lowest dose that they can administer, and they're working hard to get there. They are not going to want to give you any more than you need. The AAV, viral delivered gene therapy, it can achieve long-lasting, perhaps permanent gene modification. Wouldn't it be great if you could take one injection, have DUX4 turned off, and go ahead with the rest of your life? Yes, it would be. We don't know fully that that's what will happen. We don't know that it'll be for the rest of your life. Perhaps those muscles that receive that therapy change and die over time, and you get new muscles and they haven't had that therapy, but it could potentially be curative. Expression can be targeted to muscle. And the virus has been extensively used in other diseases. This has taught us a lot. We need very careful monitoring of a patient as soon as they receive this virus, because you can have very strong, very bad reactions to it. But if you're monitored closely, that can be managed. But you're not necessarily going to have a very strong, very bad reaction, but those have happened. So it can't be used in people who have been exposed to this virus before, because you have antibodies to it. So a person who has seen this virus, and it could be up to 30% of our population, can't be given it. You will be tested in advance to determine if you have those antibodies. As I described, there can be extreme immune reactions that can damage organs or be fatal. We already know how to carefully monitor patients if potentially for that effect. And then finally, there has been some association that the immune system, if it's suppressed, because that may be a part of the treatment, there's long-term risks of infection and cancer. And we just have to be open and honest about the risks and the potential benefits. When I asked a couple of friends who are patients to review it, they said, put it a little simply, please. Small molecules, usually oral administration, requires daily dosing. Antibodies with IV administration will require infusion at a clinical office where access could be challenging. We're going to have a hard time with getting enough IV sites, IV administration sites, once we start to have positive therapies. We're going to have to really work hard to fill that. Again, IV administration for the conjugated oligos. And then the viral delivery, Perhaps 30% of patients will not be eligible because of previous exposure to the virus, and otherwise, maybe you could have a one-time treatment, and that's pretty exciting. Just final couple of words here. And you guys I know this is the first time we've had a patient meeting in Europe, and a lot of the initial work. Great, we're done. I could done. The CTRN, a Clinical Trial Research Network, has been doing natural history studies in the US, in Europe. They're very, very, very important. They help us design our studies. We are so appreciative. I'm not going to talk too much about this. I'm going to talk a little bit more about MOVE Plus and MOVE Pediatrics. They're happening around the world in the US and Canada, but I'm showing here a number of places, and the pediatric work is happening in the US, UK, Canada, Brazil, and perhaps Australia if we can restore the NIH funding. And we still need patients to enroll in natural history studies. Please enroll. Please help us understand how to best measure if the therapy is effective. And I talked a bit about this. Finally, I want to speak to this question about genetic testing. Many people in FSHD families may not have their children be tested because there wasn't a therapy in the past. Now there is going to be a therapeutic approach within the next 5, 6, 7, 10 years, and genetic testing is essential for you to be involved in our clinical trials. So please consider, as you consider the health of your family, the health of yourselves and your children, to get tested so that you're eligible. And that's it.